Hello and welcome to my BIOL 395 presentation. My name is Gus Fridell, and today I'll be talking to you about simulation for the detection of different modes of polyploidy. I worked as a senior undergraduate in the Vision Lab, and my graduate student mentor is Brian Reedy. So to start, what is polyploidy? Well, polyploidy is a condition in which the cells of an organism have more than two paired sets of chromosomes. So we know that in diploids, there are two homologous copies for each chromosome, but in tetraploids, which are polyploids, there are four homologous copies of each chromosome. In hexaploids, there are six homologous copies of each chromosome. There are two main flavors of polyploidy, one of which being allopolyploidy. Allopolyploids are usually hybrid offspring whose chromosome complement consists of more than two copies of chromosomes derived from different species. So say species A and species B underwent some interspecific hybridization to create a new species, which is an allotetraploid. Autopolyploidy is another example of polyploidy. In autopolyploidy, meiotic disjunction or some whole genome duplication event results in a doubling of the ploidy. So you went from a diploid to a autotetraploid. So polyploidy occurs mostly in invertebrates and plants, and some of the reasons we should study polyploidy is that it has important implications in agriculture as well as species diversity. So, for example, millennia of hybridization induced by cultivators has created allopolyploid species of several extremely common agricultural staples. For example, wheat and tobacco, cotton and quinoa, peanuts, I believe, as well, and strawberries are actually naturally occurring allopolyploids. So typically types of polyploidy can be verified with methods like karyotyping, but these procedures require a lot of time, skill, and a high fidelity of specimen sample. These days, sequencing technology has advanced to the point where a researcher can obtain a whole genome even in the field or with some really easy to harvest sample like a couple leaves. Developing methods that take advantage of the ease of sequencing will allow for faster polyploidy detection. So how do we detect polyploids? Allopolyploidy was distinguished from autopolyploidy in a landmark study of polyploidy in Hawaiian silver swords. In that method, the researchers used highly conserved genes to construct phylogenies that determined the relationship between the subgenomes of the allopolyploid and their closest extant relatives. We employ the same framework in the detection of simulated allopolyploids using a highly conserved set of universal genes to judge relatedness. So, for example, you could have species A and species B, they're both diploids, and they create an allopolyploid when they hybridize. And the allopolyploid contains one subgenome derived from species A and another subgenome derived from species B. Now, the expected phylogeny of this is that species A falls out with subgenome 1 and species B falls out with subgenome 2 because those are the species that gave rise to these subgenomes inside of the allopolyploid. In an tetraploid, for example, you could have a diploid species A that undergoes a duplication event, and now you have two subgenomes. And the expected pattern for the autotetraploid may look something like this, where subgenome 1 and subgenome 2 are more closely related than the extant species A. So we start with the simulation for the allopolyploid, and we do that by starting with a haploid reference genome, and then using a simulation script written by Tosin Olainka, who previously worked in the lab as an undergraduate. Each copy of the DNA is duplicated. Random mutations are induced at each duplication. Here, colored bars represent mutations of loci of interest. After a couple more duplications and generation of mutations, we arrive at an allopolyploid and two relative species. Now, the expected phylogeny of this is that, as we stated earlier, species A, the relative species, falls out with subgenome 1 because they shared a diploid ancestor, and species B and subgenome 2 fall out with each other for the same reason. Now, if we look at the yellow bars, the relation between the subgenomes of the allopolyploid and the relative species becomes apparent. So another important element of the simulation that is under our control is that we control the amount of divergence after each split by modifying the mutation rate in the simulation. So you notice how these bars look a little longer. In this instance, we allowed for a large amount of evolutionary time, which corresponds to a large amount of divergence between the parental diploids. This ensures that the parental diploids aren't too closely related, and thus it is easier to detect the allopolyploid signature. Now, allow me a brief digression here because it's also important to consider the expected phylogeny for the autotetraploid. To recap, an autotetraploid occurs when a diploid species undergoes a duplication event, and the resulting phylogeny shows that the subgenomes of the tetraploid are more closely related to each other than to their parental diploid. Now, we are interested in exploring the limits of our predictive power to distinguish the autotetraploid outcome from the allotetraploid signature. In this case, as shown by these shorter red arrows, the divergence between parental species is very low. And since the parental diploids are not as distinct genetically, it becomes more difficult to distinguish the allotetraploid. To see what I mean, consider that an autotetraploid is simply a diploid with a duplicated genome. Now, if two closely related diploids merge into an allotetraploid, our ability to distinguish this from an autotetraploid from an allotetraploid diminishes the more closely related these two parental diploids are. We would like to determine exactly the amount of divergence required in parental diploids such that we can confidently distinguish the allotetraploid signature from what we might get if we're not so confident about the allotetraploid signature. This is a simplified workflow overview of the pipeline to go from genome sequence to phylogeny. Start with a model genome. In this case, I chose Arabidopsis thaliana, the vascular plant. And after simulation of the allotetraploid, we generate reads using ART, which simulates actual short reads as if the genomic data came from an actual specimen. We then identify variant genes and construct phylogenies analytically. In the future, I intend to work with real genomic data. The Vision Lab is interested in determining if interspecific hybridization is occurring between endemic and invasive species of guava trees in the Galapagos Island archipelago. One consequence of polyploidy is that it can result in reproductive isolation. That is, polyploids and diploids may not be compatible for forming viable hybrids. Some early genomic evidence suggests that BIOL, the endemic species of guava on the islands, may be an allopolyploid, which, if true, would mean that the invasive species of guava, shown here, and BIOL are not hybridizing, which has important consequences on conserving the native species. And these are my references. Special thanks to Dr. Vision and Brian Mancini for all of their tutelage and mentorship, as well as allowing me enough agency in the lab to work on my own project. I really feel grateful that I'm in such a great lab. And additionally, I would like to thank Tosin Alenka, who is now in graduate school at BU studying bioinformatics. He was very helpful in getting me off my feet when I first joined the lab. Next semester, I plan on pursuing a senior honors thesis in the Vision Lab. Thank you.